Hi, Stan with 3D PT. Three, two, one. Hi, Stan with 3D PT. About 2 years ago, I published probably my most popular file on Thingiverse, which was the DNA RNA Manipulative. It's on the education section of Thingiverse. So I'm getting ready to actually sell those commercially. And so I'll walk you through some of the parts, and what I've made is the display set that has holes for 3mm thread. That way you can thread it up and display it. Another option is to get just these this magnetic tape and use that as a magnet board. So I'm going to do that for a display set. And the goal is to produce a model of DNA like this. And here's what I did for a grant earlier to show a set of codons that you could actually demonstrate a certain concept with. Now this is probably bigger than what you want for a class set. For each group of about 2-3 students, you'll want at least 6 nucleotides long, just based on how much time and material you have on your 3D printer. And so this is for the DNA part. I'll do part 2 for the RNA and show how that works. Alright, once you print the pieces, I'm going to start with my phosphate pieces here. And to trim them off, you'll notice that there's some rough edges. There's some rough edges and some flash on them. Now to print this, I actually print it in 2 layers. This way the lettering sticks out. So to trim this, I could use emery board. I actually get these for free at like job fairs and teacher development training. You can use your hobby knife, pliers, or flesh cutters. And so you want to go through your pieces, make sure they're all nice and even. And no sharp edges so they don't poke your students or just get caught up in things. Alright, once you have your pieces printed up, you're going to need to show your students the different parts. So this is the deoxyribonucleic acid, and so the deoxyribose sugar is labeled D. So you're going to need a dozen pieces at least. And again, I color coded them 2 parts. So in my case, the deoxyribose sugars are going to be white on top with a brown D, and the phosphates are printed brown, and the phosphates are printed brown with white lettering. So what you could do actually to save you some time is print a layer of your DNA pieces first, then your phosphate pieces second, and just switch back and forth between them. So to start off with, have your students build a backbone with the deoxyribose sugars and phosphate pieces, and start with 3 for right now. Now, is there something wrong with this? Because DNA is not parallel, and you notice how the pieces only have the notches on one side. So DNA is actually anti-parallel, which means one strand runs one way and the other strand runs the other. So your students will have to make sure they show you that they run like this. So now you have your nucleotides laid out, you have your DNA backbone. How do they go together? Well, Chargaff's rule always says that adenine and thymine always pair together. and cytosine and guanine come together. So you can see how I can't put thymine and cytosine together with them fitting. And same with guanine and adenine, they won't fit. And so these two will always pair together in different ways. So I'm going to choose 3 of them for right now. Now DNA always goes between the backbone, so cytosine and guanine, adenine and thymine. And so your students could have fun just making random ones to start off with. And right now it doesn't matter how they're oriented, because each of these is a set of your DNA. So now I have 3 sets of letters on each side, which is 1 codon. So here's a complete codon. There are 3 nucleotides on each side. They're paired correctly, adenine to thymine, cytosine to guanine. And so you could have your students demonstrate this first and just use random ones for right now. Later on, you could develop this and actually have the students code for specific amino acids. And you notice that each of these are color-coded opposite of each other. So when you print these in 2 layers. And what I like to do is switch back and forth between printing them. That way, I start off with the adenines on a blue layer and on pink. And then when I switch to thymines, it's a pink layer and a blue layer. This color coding helps illustrate the concept of how they're paired together. This also is great if you're running low on one type of filament. This way you can kind of stretch it further. The color coding also provides visual reference, so that way you could see how they pair, because obviously, blue and purple don't pair together in this case. Make a set of at least a dozen D's and P's, and that'll give you enough for about half dozen of each of these nucleotides per group. So I'd recommend group sizes of about 2 to 3 students, no more than 4. So here's 2 codons worth of DNA, and again it's running anti-parallel. There's a 3' and 5' file as well, so you could show those in high school class to show where the 5 and 3' ends are. And right now we have all the things paired up, guanine to cytosine, adenine to thymine. Now what if we want to make RNA off of it? So what happens when we make RNA is that this actually splits in half. So carefully have your students pull this apart. And it may help to string these together. And I'm going to remove the 3 and 5' right now. Yes, your students will make a little bit of a mess of this, so don't worry about that. These are pretty big pieces. I designed them specifically so you could easily see them. So the first step is that the DNA will divide in half. There's an enzyme that comes along and splits that apart. And you're going to use these RNA pieces to come along. So here I did them green. Now always remind the students that instead of thymine, uracil replaces that. So I did make the uracil and thymine pieces the same shape, but always pair the RNA colors together. This helps with differentiation, makes it easier to sort. So start by making the ribonucleic backbone. So it still has a phosphate between them, but this time you're going to use the ribonucleic ribo sugar. And you want to again make it the same length as the DNA piece you're trying to match. So let's see, 3, 4. That's 6 with the phosphate on the end. And to bind it, I'm again going to use cytosine. Uracil. Guanine. Adenine. Uracil, because in RNA it always uses uracil instead of thymine. And one last guanine. And so you can talk about enzymes and how those bind together, and you could also show the differences, because in RNA, instead of thymine, you always use uracil. And then this gets split off by another enzyme to become messenger RNA. And once you have this messenger RNA, you notice that it's similar to the DNA that it read off of. So what's going to happen is each of these is one codon. Each 3 is a codon. And you're going to read each codon with a messenger RNA piece. And what happens is this comes along and reads, the transfer RNA reads this, binds an amino acid to it, and then builds that amino acid as it goes along. So in the next set of codons, it adds another amino acid, and you keep building this chain out of amino acids to make your protein. And if you use the expansion set that's also available, you can actually code it for the individual unique amino acids. So that's DNA and RNA manipulatives. You could go to my site at 3dpt.club for more information. Also, I have some worksheets and lesson plans available on my TeacherPay teacher site, which will also be linked below. If you'd like to buy a set, I will start selling a magnetic set. So for those, I'm just going to put some of those flat magnets on the back. And those will be up on my Etsy page as well. Please subscribe, ring the bell for notifications. I will try and publish more videos about my education projects, as well as cement printing and other things. Thank you for watching, and have a nice day.